Hello, everyone. I'm Dr. Andrew Kaufman, and today I'm here with a special report on monkeypox. Yes, you heard it right. There's been a lot of hype around this new monkeypox virus, which is really not very new at all. Now, in the past, when I have done all the research with respect to SARS-CoV-2 and showed that it actually doesn't exist, I also said that other viruses that I've looked at don't exist. And I emphasize the importance of that so that when a new scare or threat comes out, that you won't be fooled again. So now here we are with the next fake virus threat. So today I'm going to tell you all about the monkeypox virus. First, I'm going to start off reviewing some of the hype surrounding it and all the scare tactics and what the governments have done so far. And then I'm going to go into the isolation papers to answer the question definitively, does the monkeypox virus actually exist? And then finally, I'm going to actually explain an alternative mechanism of how the type of disease with pox lesions or skin vesicles can occur without any virus, but by exposure to toxins, a mechanism of disease that would be consistent with terrain theory. So let's get started. Okay. So first, I'm going to play you this video from a news station in Texas, which gives a little bit of the background of what's going on. So let's listen together.
Public health officials are watching closely for the potential spread of monkeypox in the U.S. So far, the CDC has confirmed one case in Boston. There are suspected cases in Broward County, Florida, and New York City.
This should be taken seriously.
But Dr. John LaPook says there are huge differences between monkeypox and COVID-19 that has killed millions worldwide.
We've known about monkeypox for more than 60 years. There are vaccines. There are therapeutics. We have a lot of experience dealing with it.
Monkeypox is a virus found in wild.
Now, let me just intervene for a second because I looked on the CDC's website to see what treatment they do have available, and they don't really have treatments available. In fact, they state that there's no individual treatment that is available. However, they do use smallpox vaccines and immune sera from smallpox occasionally, and they say this is for the purpose of containing a threat within a community rather than for treating the individual, which is kind of an interesting distinction, but that's what the CDC says.
Animals like rodents and monkeys. When it infects people, symptoms include fever, headache, aches and pains, fatigue, and swollen lymph nodes. Within several days, a rash develops that progresses to raised blisters.
Now, I'll let you know also that in many of the studies where they looked at this, where they did the viral isolation, they actually did not have these symptoms.
That turn into scabs. The illness usually lasts 2 to 4 weeks. The patient in New York City is being isolated and treated as positive while waiting for final confirmation from the CDC. During a 2003 outbreak of monkeypox in the U.S., 47 people became ill, but nobody died.
We've had this monkeypox in larger numbers in the past.
While traveling in Asia, President Biden said the U.S. has the necessary tools to fight the virus.
I just don't think it rises to the level of the kind of concern that existed with COVID-19.
Health experts say the virus can be transmitted through body fluids, skin, and respiratory droplets, but it usually requires close personal contact to spread. Elise Preston, CBS News, New York.
Sophie!
Okay, let's get rid of that. Okay, so now you get the idea. They're comparing it to COVID. They're saying that it's not as severe, but on the other hand, they are raising concern. They put in the pictures, the scary picture of the hands with the lesions. So let's now shift and look at the CDC website here about monkeypox. Now it says it's a rare disease. It's from the orthopox virus genus, so they say. And if we look at the second paragraph here, it was first discovered in 1958 when 2 outbreaks of a pox-like disease occurred in colonies of monkeys kept for research, hence the name monkeypox. So what this says is this is monkeys in captivity, not monkeys in the wild. The first human case of monkeypox was recorded in 1970 in the Democratic Republic of the Congo during a period of intensified effort to eliminate smallpox. So we're going to look at that paper as well. Since then, monkeypox has been reported, blah, blah, blah. Now, of course, we have all heard about the new monkeypox cases. So let's look at monkeypox in the United States here, one of the tabs on their website. And of course, they're talking now about the current person in Massachusetts who is returning from Canada. And there are some prior cases you see November 2021, July 2021. So even during the COVID pandemic, we've had a couple of cases reported and some more information. So not too much, no massive casualties or anything like that. Now, of course, they recommend that this is how you can test someone. And they specifically mention real-time PCR. From a lesion is the testing. And here is how you can actually order it for more information about the PCR test. So, of course, we have a situation where if there is no virus, there can't be a test. So we won't look specifically at the PCR studies until we make that determination. Now, I want to point out that some action has been taken. So according to Newsweek, the U.S. buys monkeypox vaccines. And if we scroll down a little bit, we see that there has been, a contract with Bavarian Nordic amounting to $299 million, which would provide 13 million freeze-dried doses. So the government is already buying lots of doses of vaccine. Okay, sound familiar? All right, so now let's shift a little bit and look at the papers that we just heard about from the CDC. So this is the original 1959 paper, A Pox-like Disease in Cynomolgus Monkeys by Magnus et al. And so this has been referenced in all of the papers. And this has to do with the monkeys in captivity. Now, interesting here in the introduction paragraph. So during the summer and fall of 1958, 2 outbreaks of a non-fatal pox-like disease in cynomolgus monkeys have been observed in the monkey colony in this institute. Both outbreaks occurred rather late after the monkeys had been received, 51 and 62 days after arrival. And only a small percentage of the exposed animals showed signs of illness. I think it was somewhere around 6 animals total. But since this took place, almost 2 months or just about 2 months after the monkeys arrived at this institute, one has to wonder, did they not get sick at this institute? How did they keep them? What did they feed them? What drugs or inoculations did they give them? Those things could have caused the stress in some of the monkeys to produce the disease, but this was, of course, never looked at. Now, I'm going to skip this part of the viral isolation. Well, actually, let's not skip it because it gives additional information that's not shown below. So if we look here in the highlighted portion, this is describing the isolation experiments. So they did 2 types of virus isolation. They did egg inoculation, which is where they take the fluid from a pustule and put it in an egg that's developing. And then they also did the tissue culture experiments that I've talked about quite extensively. Now, in these tissue culture experiments, it says that the pustules or the material was collected by putting a cotton swab in the pustules and then putting that in bactotryptose. And then that contains penicillin and streptomycin, so 2 antibiotics, one of them being especially toxic to the kidneys. Then they did some mild purification with low speed centrifugation. And then they put that either on the chorioallantoic membrane of the chicken embryos. And then they also put it into tissue cultures of monkey kidney cells, human amniotic cells, and HeLa cells, which is a cancer cell line that you might have heard about. I think they made a movie or a book about it, Henrietta Lacks. And they said a total of 3 virus strains was isolated. Okay. Now, let me go down a little bit further. So the typical isolation experiments of the day. And then here, I want to read this part, electron microscopy, because we're going to look at that as well. So they say what kind of electron microscope and the magnification, but they actually have 2 different samples. So the pus-like material from pustular lesions was removed, placed on and allowed to dry. So, and then the grids were treated with trypsin solution. So they actually, interestingly, they took the pus itself and put it under the electron microscope, not the fluid from the cell culture. So this is definitely different, but they digested it with trypsin. So trypsin is a serine protease. It's a type of enzyme that digests proteins. Now, I'm not sure what the rationale for this is, but it certainly affects what's in the sample. It could be to break up some of the pustular material, like from the immune system, but it's non-specific. And then fixation was then after that, they washed it in distilled water. Now, that's kind of important because distilled water has no solutes, so it could pull solutes out of the proteins in the cellular environment, and this could cause the proteins to be denatured, and it can mess up what is going on with your sample or how it looks. And then fixation was carried out with the vapors of 1% osmium tetroxide for 15 minutes. And then the grids were heated at 70 degrees C. Now that's quite hot. The body temperature is 37 degrees C. So that's definitely, like 150 degrees, something like that. Prior to shadow casting with platinum. Now that's important. I'm going to talk a little bit more about what that is. And then the egg-adapted virus grown on the chorioallantoic membrane. So that means this is the pus that was put into the chicken embryos, was also purified purified, it should be in quotes, for electron microscopy by differential centrifugation and precipitation with citric acid, according to the methods of Henderson and McLean. The final suspension was placed directly on Formvar-coated grids, dried and further treated, as stated above. So that's talking about with the platinum and the osmium tetroxide, with the exception that no digestion with trypsin was carried out. So this sample didn't have the trypsin digestion. Okay, so we'll go down a little bit. And this picture shows the lesions on the monkey. Once again, the monkey lesions. This is in the embryo. Okay, so once again, now we go back to the virus isolation. Okay, it was attempted only from 1 monkey during outbreak number 1 and from 2 monkeys during outbreak number 2. So we have 3 animals that underwent this isolation. Now, there actually are some controls in this experiment, so we'll get to those later. Now, here is where it shows the cytopathic changes. So it shows the cytopathic effect, which they say, of course, is the proof of the presence of the virus. Sorry, there's a lot in this. Okay. So they use the typical tissue culture experiments to show that they had cytopathic effects, and they also showed lesions in the embryos. which they have shown in the other type of virus experiments, because they're putting toxic materials in. Now, another experiment they did is that they took the different materials. Okay, some of them were from the tissue culture, I believe, at different dilutions. And then also, I think that they, let's see, what did they? Yes, they just used dilutions of the so-called isolated virus. And then they put them in experimental animals. So they did it by intradermal inoculation, where they put a needle and injected it under the skin between the layers of the skin. And then they also did scarification, that's where they take a scalpel and make multiple incisions superficially in the skin and then put the material on that. Now, of course, these animals developed lesions where they were injected or scarified. Okay, but they didn't develop any systemic disease. So in other words, they put toxic soup from a cell culture directly into their skin by making an incision or injecting it, and then they got lesions on their skin at the site that they did that. What a surprise. They also, let's see, what did they do? I think they did some more. Yes, they also put it into the eyes of rabbits, and they put it into the brain of adult mice. And of course, when they use the toxic material from the embryos with the intracerebral in the mice, and of course, those mice developed encephalitis and died. They also, in suckling mice that were only, I think, 2 days old, yes, they were inoculated intranasally by a method described earlier. Well, that is putting it essentially under pressure. Now, this is a, tiny little baby that doesn't have all of the skull bones fused together, and putting it under pressure up the nose. Some of it probably got into the head and the brain. And of course, that killed the animals as well. So not very surprising, nothing specific about a virus here or how it would spread in nature. Now, these are the images here, and we'll go to the caption so we can see exactly what's what. But those are the supposed electron microscopy images. So figure 11 are from the pus-like material, and figure 12 are from the partially purified suspension of the eggs. Okay, so that's this one here at a higher magnification. And I'll go back up, and I have a bunch to say about these images. So, okay, so you can see the general that there are these sort of ovoid, hexagonal type shaped white areas. Now, this is done by the technique that I showed you, or I told you about, known as, sorry, I have some. Platinum shadow casting. So that is a technique used to improve contrast, and explains how it works. And essentially, where the areas that do not take up any stain show light. Okay, so that there essentially could be gaps in the slide where there are white areas and there's no material that takes up the stain of the platinum. So the white areas essentially could represent nothing, or areas where there's actually no biological material or less biological material. That's quite interesting in and of itself. And then if we look here, I found this article that platinum forms nanobubbles when it oxidizes. So essentially with this technique, they're spraying down a layer of platinum and they're putting it on top of infected, diseased biological material, which most likely has reactive oxygen species in it, as well as being mixed with lots of acidic or toxic reagents during the preparation process. So these could potentially oxidize the platinum, and that's all we're seeing on those slides. Now, I also want to point out this image here, because as we saw from the news video earlier, this is the image that's shown everywhere. Okay, I did a reverse image study actually to find this, and it was in, every news site, the New York Times, the Washington Post, et cetera, and all kinds of other websites, but not anywhere in a scientific study. So I was able to find it on the CDC website here. But interestingly, it says that it's depicted a monkeypox virion. Now, that's just supposed to be one particle, but I don't see any single particle pointed out here. Obtained from a clinical sample associated with the 2003 prairie dog outbreak. It was a thin section image from a human skin sample. Now, there's no reference here to a paper. There's no description of how this was obtained other than it was from a human skin sample. So we really don't know anything about this image, but the interesting thing is that it doesn't look anything like these images from the original publication. So the shape is not really consistent because they describe these as having a typical square shape. And I can show you that in the text, right? That they say this is essentially like a rectangle or a square. And then here you can see that they're very rounded, right? They don't have that boxy appearance at all. And that's supposed to be one of the visual characteristics to differentiate them. So once again, we're at a situation where essentially it's really impossible to know what we're looking at here. Okay. Based on these images. So let's go back to this paper. Okay, now this is the really interesting part. So presence of monkeypox viruses in tissue cultures prepared from kidneys of apparently healthy monkeys. Now, this kind of reminds me of the Kidney 360 paper where they found particles that were identical to SARS-CoV-2 particles. So here, during the first outbreak of the pox disease, some apparently healthy cynomolgus monkeys, which belonged to the affected shipment, had been sacrificed for other purposes. The kidneys from these animals were trypsinized and culture tubes were prepared from the cell suspensions. In some of the tubes, approximately 5%, cytopathic changes developed 7 to 12 days later, and monkeypox virus was isolated from these cultures. This is amazing. During the second outbreak, monkeypox virus was again present in a small percentage of the tubes prepared from kidneys of apparently healthy but exposed animals. So right here, this is the control experiment. We found the same exact finding of cytopathic effects that was identified here definitively as monkeypox virus from animals that did not express the disease at all. So that means whatever we found, it's not related to this disease. And I would say that we can't say that it's a virus because we don't really have any information. All we have is that a mixture of things from these pustules was poisonous to these cells in some way without any control, without a biological sample. So we don't know that the cytopathic effects might have occurred anyway. So all we have is that. There's no evidence of a virus anywhere in this experiment. Okay, now, so this is from the discussion section, and I believe this is the this is the part that actually says what they found here. Say, the present study reports 2 outbreaks of a non-fatal pox-like disease in cynomolgus monkeys housed in this institute. The clinical manifestations observed in the monkeys were so typical that it seemed fairly certain that the disease was caused by a pox virus. So in other words, they had pustules, and that was so typical of these pox virus diseases that it must mean it's a pox virus disease because nothing else in the world causes pustules. Not true at all, of course. This assumption, exactly, it was an assumption, was substantiated by the isolation from the pustules of an agent which grew readily on the chorioallantois of chicken embryos, producing characteristic focal lesions. However, that also the kidneys culture specimens from the healthy monkeys also caused this problem. Morphologically, the agent was found to have the size and brick-shaped appearance typical for members of the pox virus group, except when the CDC isolates them and take images of them. Then they're not brick-shaped at all. Furthermore, the antigenic relationship between the newly isolated agent and vaccinia virus could be demonstrated by neutralization, hemagglutination, inhibition, and blah, blah, blah. So that means that it gave you the same results as the smallpox, as the vaccinia virus, which is the used to make the smallpox vaccine. So in other words, it's not unique. So here we have a whole lot of nothing being discovered. Okay? So let's go on to the next incident, because this has to do with the first human case that was reported in 1970 in the Democratic Republic of the Congo. Now, we have two papers from the same journal that describe this situation. So this is from the bulletins of the World Health Organization in 1972. And so this paper presents clinical and epidemiologic information. So I'm not going to go into it in detail, but it said this patient was the first recognized human monkeypox case in medical history. Okay. And then I'll just give this little description here. On the 1st of September, 1970, a 9-month-old child suspected of having smallpox was admitted to this hospital in the DRC. Specimens from the patient were sent to the WHO Smallpox Reference Center, Moscow, and a virus similar to, if not identical with, monkeypox was isolated. Now, that does not sound very conclusive. It's either similar to, if not identical with. So they can't really tell, is it similar, is it identical, is it not identical? Okay, I thought that was very fascinating. Now we go to the paper, where they actually supposedly did this virus isolation. And this is another long paper here. And what they did was that they say they isolated this monkeypox virus and they call it Congo 8. And then they compared it with other viruses isolated from similar cases of illness, and to see how the results compared. Okay, so, all right, now materials and methods here. So material from the patients in the DRC. Now, I thought there was just one, but they write patients, was obtained through the World Health Organization by air in a special packing consistent of the contents of skin lesions, so scabs and smears on slides. Now, they didn't say anything about how they prepared that sample in here, but they did say the type of cytopathic effect was determined in the following cell cultures: 2 primary chick embryo fibroblast and monkey kidney cells, the old Vero cells, and 4 continuous more Vero cells, A1, PEK, and HEP2. Okay. So essentially they did, and now there's no electron microscopy in this, but here we have the cytopathic effects in panel A. And interestingly, in the smallpox strain that they used as a comparative test, there were no cytopathic effects. So why would that happen? Because supposedly smallpox is more virulent. Now, they, of course, just want to say that it distinguishes them, but you have the same thing here in a different cell line, that you have more cytopathic effects on the panel of of this Congo 8 specimen. So once again, though, all we have here is cytopathic effects in a culture with no control study or no control sample. And there's a lot of antibody tests, which I'm not going to go through because we know antibodies are not specific. But here's in the discussion section, I want to go through this with you and show you what they actually said. So here, experience gained in recent years in studying variola virus has shown that it is distinguished, like a number of other viruses, although to a lesser degree, by intraspecific variability or natural variation. Now, I'm not sure what this means exactly here. Is that smallpox is different. When they do different experiments, they get different results, but so they're saying that that's a property of the virus instead of saying that they didn't really do scientific experiments. All right, it goes on. The differences found were related to the degree of pathogenicity of the strains for chick embryos and white mice, the sealing temperature for pox development, and some other indices. Now, what that means is that they took the fluid from these cell cultures and they injected them in white mice and in chick embryos, and they got bigger lesions with some of these things and smaller ones with others. That's what they're talking about. And it said, however, not one of the strains of variola and elastrum viruses investigated proved capable of causing the development of lesions on scarified skin in rabbits. Now that, of course, contradicts results of previous experiments with variola. The Congo 8 strain isolated from the patient AI with an illness resembling smallpox caused marked specific reaction when it was placed on a scarified rabbit's skin. So in other words, it caused a big skin lesion when they cut open the skin and then put this poison soup on it. This feature of the Congo 8 virus, combined with other characteristics, the hemorrhagic type of pox on the chorionic allantois membrane, so in other words, the chicken embryo, the types of plaques in the tissue culture, so what we saw above, high degree of hemagglutinating activity, that's clumping up blood, sealing temperatures for the development of lesions. So all of this stuff just means that it's more poisonous than the other samples. That's all it means. It says nothing specific, but it makes it impossible for it to be considered a variant of variola because it's so much more poisonous in these experiments. On the other hand, the properties of Congo 8 proved to be indistinguishable from those of the classic representative of monkeypox virus, the Copenhagen strain. Well, that's as poisonous and provided good grounds for identifying it as a monkeypox. Good grounds. Yes, good grounds, not conclusive evidence or anything like that, just good grounds. The evidence thus indicates that the monkeypox virus can cause disease in man. So they went from a really loose argument to saying that it can cause disease in man. So there's nothing specific here that they have to identify it. There's no specific test. There's not even a specific antibody. There is no control experiment where they have other material besides these lesions from sick animals to compare. And so they can take this poison and inject it in the skin and cause a skin lesion that's not exactly causing the disease, because the disease, like we learned about, starts off with some general malaise and fever and then progresses to swollen lymph nodes. And then after that, there's an eruption of a rash. None of the animals got that. So where is the specific proof that what we have here is a virus and that it causes any disease? Once again, we come up very short. Now I'm going to just go briefly into one more paper, because this is the first case where they supposedly found this virus in a wild animal, because what they're saying initially is that they found it in captive monkeys. And of course, that could mean, and likely was because of the long delay before symptoms, that it was caused by some condition in captivity. Whereas when they identified a human case, they said, well, there must be a reservoir of it in the wild somewhere. So they started looking to animals to see if they can find it. And so this is the first time that they say they found it in nature in a squirrel, of course, not a monkey. They say the monkey thing was just an accident, of course. But I'll read, in July 1985, a joint government of Zaire, World Health Organization epidemiologic team did a survey in northern Zaire looking for human cases and collecting animals within 40 kilometers of Yambuku, Bumba zone. And in Yambuku, they had an outbreak of Ebola hemorrhagic fever in 1976, an unlucky place. There must be lots of industrial toxins there. So here we have one of 383 animals sampled, a squirrel, found 300 meters from a village where a human monkeypox patient had been recorded in 1981, 4 years prior, had skin eruptions. Wow, so they found one out of almost 400 monkeys with skin eruptions, and it happened to be a squirrel. So they took serum skin and organs from this animal, were tested by 2 World Health Organization reference laboratories, namely the pox virus laboratory at the CDC in Atlanta and the Laboratory of Smallpox Prophylaxis Research Institute for Viral Preparations in Moscow. Both laboratories isolated monkeypox from the skin and lungs, spleen and kidneys. Wow, all over the place. The orthopox virus antibody titer, blah, 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 and monkeypox specific antibodies were identified in serum by radioimmunoassay absorption test. The virus was also tested by monoclonal antibodies and found identical to monkeypox virus from an infected patient. This is the first time monkeypox virus has been isolated from an animal infected in the wild. Now, interestingly, there's no reference to any of these experiments. They must not be published. They're not listed here in the text or at the end of this brief article, even though they were done in supposedly two different laboratories. Now, those antibodies also, you can see there are 3 references here, but they are not those references. So antibody tests are not really specific, as we've seen many, many other examples. So we're not going to go through even taking that seriously, but this isolation, who knows if it has really been done. I want to now that I've reviewed the evidence, and I think we can come to the conclusion that there has never been a monkeypox virus that's actually been discovered by any credible scientific evidence. We have the typical faulty isolation and electron micrograph images here as usual. And we have to ask, why is this coming up now? What could be the plan? So I refer you to, you might have seen this, but similar to Event 201, there was another tabletop planning exercise conducted in March of 2021, of all times, so during the COVID pandemic. That was a joint venture between NTI, which is the Nuclear Threat Initiative, and the Munich Security Conference. Now, these are both globalist organizations. The Nuclear Threat Initiative has Ted Turner, for example, on the board, lots of big executive business people, government officials, ambassadors, people like that. And similarly with the Munich Security Conference, I looked at a couple of their board members' Twitter sites, and you saw references to the Rockefeller Foundation and other globalist organizations. So this is what they're after. And so I want to just point out a couple of quick areas in here on page 8, first of all. Okay, here. So this describes the exercise scenario, which portrayed a deadly global pandemic involving an unusual strain of monkeypox virus that first emerges in the fictional country of Brinia and eventually spreads globally. Now, this is the interesting part. Later in the exercise, the scenario reveals that the initial outbreak was caused by a terrorist attack using a pathogen engineered in a laboratory with inadequate biosafety and biosecurity provisions and weak oversight. The exercise scenario concludes with more than 3 billion cases and 270 million fatalities globally. So quite interesting that this is the lab-created virus scenario here actually materialized into a tabletop exercise. And you could see where this leads to an emphasis on biosecurity as one of the main initiative measures. And so that has to do with tracking and tracing, vaccine IDs, all of that kind of stuff. And I want to show one more, because these are the findings from this exercise. So in other words, what they're going to recommend based on this exercise. And here we go. Governments should improve preparedness by developing national level pandemic response plans built upon a coherent system of, quote, triggers that prompt anticipatory action on a no regrets basis. So in other words, they want you to predetermine some trigger, and they want it to be anticipatory. So in other words, they want you to get ahead of the game and act early. So create fear and panic, act early to prevent, of course, the potential destruction that they say would come. And they say to do it on a no regrets basis. In other words, for leaders of nations who have to, take away people's freedoms, shut down businesses, quarantine, all of this kind of policy, which is very, very unpopular. They are supposed to do it without regret. So, here's a quote from one of the participants on the panel on the side here. It will be chaotic and frightening, but you can't wait until you have certainty. You have to act with no regrets. That's right. We definitely don't need certainty before we start taking people's freedoms away. So this could be, of course, where we're heading. Hopefully that will not be the case. But I wanted to spend a little time here first just talking about what could really cause this type of an illness so that you will not freak out if you actually see someone who has some kind of pox illness. So I just want to show you here, this is from the National Cancer Institute. And I think that this is eventually going to be talking about melanoma, but they do a very, very nice job here of explaining how the skin is structured. And you see there are different layers of the skin. And actually, I looked at this picture before and I thought, wow, it kind of looks like a forest. But in any case, you have different layers. And I wanted to point out here that the outermost layer here, the stratum corneum, is made up of 10 to thin layers of continually shedding dead keratinocytes. And these are all kind of stuck together in a mass. And I want to point just to this collagen and elastin here. So the dermis is held together by a protein called collagen made by fibroblasts. Fibroblasts are skin cells that give the skin its strength and resilience. Collagen is a tough, insoluble protein found throughout the body in the connective tissues that hold muscles and organs in place. In the skin, collagen supports the epidermis, lending its durability. Elastin, a similar protein, is the substance that allows the skin to spring back into place when stretched and keeps the skin flexible. So you see that there are these structural proteins that give the skin its properties of sticking together, the different layers adhering to each other, and of the resilience and elasticity of the skin. Now, when we have pustules or vesicles, and these are essentially fluid-filled lesions on the skin, what happens is that the layers of the skin separate from each other, and then the fluid fills that space. And that's what you have. And when you have the pustules, with the pustular material, the kind of whitish, thick material, that is an infiltration of white blood cells. And sometimes you have clear fluid in the vesicles. They call them vesicles instead of pustules. And that could be free of cells or just fluid with proteins and electrolytes in it. And so in this diagram, you can see that it shows there is collagen that is anchoring the layers together and forming the structure within the layers as well. So that is disrupted when you have these pustules or vesicles. So what could disrupt the collagen or weaken it to allow these things to occur? So I found an article from Wikipedia, and I'm just kind of following the trail and putting two and two together of what can cause collagen loss or damage to the collagen. And lo and behold, we see here oxidative stress. Okay, now oxidative stress is really what we have when we have diseased tissue and we have exposure to toxins. Many of the toxins cause oxidative damage, which initiates a chain reaction of free radicals, and these are also sometimes known as superoxides or reactive oxygen species. And we have, our body makes things like glutathione, for example, and superoxide dismutase, which is an enzyme that can quench these and prevent further damage. But we're talking about in a disease state here, where these things are active. So could we have a toxin causing oxidative stress, breaking down and weakening the collagen, allowing these pustules or vesicles to form? So I thought, wow, you know what? There is a disease model for that. And this is it, toxic epidermal necrolysis. So this is, it can be extremely severe, where much of the skin is affected over the whole body. And it can have a high mortality rate of up to 30%. And essentially what happens is that vesicles and pustules form on the skin, and then they break off, leaving exposed skin because it damages the top layer that separates off. So it could be like someone with severe burns. Now, interestingly, if we look here, etiology means what's the cause of it. TEN can be induced by medications, infections, vaccines, or even idiopathic. That means they don't know the cause. So medications and vaccines are on there. And in fact, reaction to certain drugs is by far the most common cause, it goes on to say, and it lists a whole bunch of different types of medications, antibiotics, like sulfa drugs, chloramphenicol, which is still used in a lot of developing countries, penicillin and quinolones. Quinolones are very, very toxic. They're known to cause connective tissue disease problems like people's tendons popping and snapping, which is a similar type of issue, but not in the skin, obviously in a tendon. Seizure drugs like barbiturates, carbamazepine, which is Tegretol, phenytoin, which is Dilantin, valproate, Depakote, sorry. How could I forget that one, being a former psychiatrist? And lamotrigine, which is Lamictal. Also non-steroidal anti-inflammatory drugs, especially oxybutazone and piroxicam. Those are prescription non-steroidals. And interestingly, antiviral drugs it lists, and allopurinol, which is a drug for gout. So a lot of drugs, and one can imagine there's probably other chemical toxins that could be present in the environment that can lead to this as well. And then I just want to show you a couple of images so you can see. So this is toxic epidermal necrolysis after the vesicles have essentially popped, leaving the skin lesions. And this is an image of monkeypox. Also, same thing, after the lesions have popped, you can see the raw skin lesions. So I think these, look pretty similar, such that you could easily confuse one for the other. And we have now a potential alternative cause of this kind of pustular, vesicular skin lesion type of disease. So if you see this, you know that this could be also caused by a toxin. So if there's an outbreak in one area, for example, there could be exposure to a toxin there. Just in case we see something like that, I'd like you to be able to interpret that without fear and be able to look at the situation more rationally. So just to review everything I've discussed with you today, we saw that there is a lot of hype and fear, and even some policies going on with respect to this monkeypox virus, that there have been a few cases reported around the world, including in the United States. The United States government has bought up a bunch of vaccine already. We know that they're using a PCR test, which couldn't possibly be accurate because there is no virus that has been actually identified or shown to exist from which they could make a diagnostic test for this alleged virus from. So once again, we have the same kind of scientific misperception or perhaps even outright fraud. And we have commercial exploitation by companies making diagnostics and therapeutics for an imaginary virus. And we see that the globalist organizations have planned at least to consider this particular virus, and they have a whole story. So if we see it escalating, we could see that story being played out to some degree. But I hope that after seeing this, you will take a deep breath, a sigh of relief, and realize that there's no such thing as a virus that causes disease. And whenever these new threats appear in the mainstream propaganda media, that you don't need to worry. And remember, you can always find a friendly face right here. Thank you.
Introducing a powerful dietary supplement. Over 1 million years in development. A highly concentrated, complex, natural formula, rich in trace minerals as well as fulvic and humic acids. Mother Nature's ancient elixir for energy, revitalization, and long life. Shiljit Mumio means conqueror of mountains, destroyer of weakness. Take in the nutritional wisdom of the ancient past. Unleash the vibrant, healthy being inside. Become the conqueror.